So Dr. Vikas Tige works as Scientist E and heading the National Center for Preclinical Reproductive and Genetic Toxicology at ICMR National Institute for Research in Reproductive and Child Health, a premier institution of Indian Council of Medical Research, Department of Health Research, Government of India. He is a veterinary graduate and a doctorate in animal biotechnology. Dr. Dige has more than 2 decades of experience in animal experimentation, mainly the drug development process, and has undertaken more than 30 projects wherein efficacy, safety of the novel molecules has been undertaken following the OECD ICH guidelines. He is having expertise in animal model development for PCOS, diabetic hypothyroidism, oligospermia, etc. His current area of research is molecular toxicology and novel drug delivery. Apart from managing the service mandate, he is guiding PhD students and postgraduate fellows, and he is instrumental in human resource development in the area of preclinical reproductive and genetic toxicology. He has shouldered the responsibility of conduction of training workshop and organized national and international conferences in the area of laboratory animal science, clinical pharmacology, etc. His current area of research is molecular toxicology and novel drug delivery. He has more than 40 publications in high impact factor journals, peer-reviewed journals, and has received many awards. Looking into present scenario of lumpy skin disease in livestock and losses occurring, it's very important to restore the livestock production. So today's topic is very much related to enhance production in livestock sector. So I invite Dr. Vikas Dige to deliver the lecture on biotechnological inventions in enhancing livestock production. Dr. Vikas Dige, please.
Thank you, ma'am. Okay. Good morning, friends. At this outset, I'm really grateful to the organizing committee for giving the opportunity to share some of my experiences and the knowledge of biotechnological interventions, which has helped in enhancing the livestock production. Today, I'm going to just unravel the journey, how exactly the biotechnological area or biotechnological tools has paved the way for the development of or maybe improvement in the livestock productions. As ma'am rightly said, this is like being my alma mater has graded in biotechnology. So I'm trying to just justify the knowledge which was acquired during the graduation, post-graduation, and the doctor degree, as well as having the affections toward this very important avenues of the government, the GDP of government of India. So I'm unable to put these slides. I think our slides has
already your first slide is been visualized. It is visible.
Not,
first slide.
Yeah. No, my slides is not moving. Are anybody is controlling from their end?
No.
No, sir. No.
Just a minute. Huh? Yeah, now it is coming out. Sorry. Yeah. So yeah, this for the those who are newcomers in this area, or maybe some of students may be attending this particular workshop. So the biotechnology is an integrated application of scientific and engineering principles in
sorry to interrupt. Here, first slide only.
Yeah, first slide only. Oh,
yes.
you're not getting the second slide?
No, sir.
I don't know why. Just a minute, just a minute. Yeah.
Dr.
Now it's
Vikas?
okay.
Yeah.
Now it's
Now
okay,
it's
sir.
okay. Now it's
Yes.
okay.
Yes, sir.
I think still there is a issue because now I lost my control.
डॉक्टर रीशेअर करा नाहीतर एकदा बंद करून पुन्हा रीशेअर करा चालेल.
That's good. No. Helpful for the animal welfare as well as the human health. So there is always a need of sustainable development by improving the livestock productions for influencing animal health, nutrition, reproduction, genetics, and breeding. So I just without wasting time, just straight away go through the journey. The first foremost area which biotechnology has paved the way for animal reproduction is synchronization of the hormones. The hormone cycling the cattle, especially by this has helped into the optimizing the timing and cost. By gathering the artificial insemination at one go, it is this particular technology is reducing the time between two calving, and then the unseasonal lactation for getting the higher price or milking can be actually planned very well. And also this technique, synchronization of hormones, has helped to avoid the transfer of sexual pathologies as well. And then the artificial insemination, the selection of the semen, the bull with high genetic potential was selected based upon the pedigree data as well as their the characteristics, and then we could able to save the store the semen, and then the better profitability of the semen. Actually, the one bull which is having a high pedigree or high quality, so one can make almost 1,000 doses from one bull, and there is no distance. There is a limitation of distance between male and female because there is no need of taking the sire or the mating studies in person to that. So the AI, the straws can be transferred from one place to another place, and then one can actually help in the development. So the very important area recently, and it is being considered because as the X chromosome of the female sperm, especially when we selected, which is always higher in the DNA percentage. That more 4% more DNA is there. So this particular technology has resulted into by using the flow cytometrical data, we could able to flow cytometric technique and centrifugation force, we could actually the heavier semen which is having female characteristics can be considered, and that can be isolated because this particular female chromosome is positively charged, and one can use this one. And this sex prediction of the calves was almost 85%, and 95% accuracy was resulted because of the semen sexing. And this is used for the enhanced progeny testing program, because the progeny testing is a very important area for sustainable livestock production. So this particular semen sexing program has resulted in the semen sexing presenting. Then they increase the breeding male production also possible because of semen sexing. And then the incidences of the sex-linked diseases was able to reduce because of this using the semen sexing technology. And we could able to save the or conserve the superior and rare animal species by using the semen sexing procedures, and then one can store the semens which are of high quality. So as this particular chart showing you that the maximum percentage of the DNA content of this goat has maximum higher DNA percentage in X chromosome as compared to other species, and these are the characteristics which helps to actually to develop the experimentation or maybe doing the semen sexing. So this particular data is useful. Then another new area has resulted into sperm transcriptome, because earlier we were concentrating only on the female part, but the sperms, which is very important component of the zygote formation. The sperm deliver the paternal genes to the oocyte and also carry the remnant messenger RNA arising out of the spermatogenesis. And the new areas like microarrays, next generation sequencing, proteomics, which could able to study the sperm RNA expression profile analysis and polymorphism in detail. And then the differences in the expression profile of these transcriptomes, especially the high fertility bulls with the higher concentration of transcriptome for membrane and extracellular space proteins location as compared to the low fertility bulls, was able to possible because of using these sperm factor transcriptomes. And these transcripts include mainly, I can spell out some of them: the protamine 1, the casein B2, and the thrombospodin receptor, then CD36 molecules. Like these are the important protein molecules which are actually important in fertility regulations or fertility developments can be possible. And then these studies have showed paved the introducing the transcriptome development. And then also here's as a sub the fertility markers. So these are the technologies being used. And then the another very important area because of the luciferase magnetic nanoparticle composition. This can be utilized for partially used for the spermatozoa detection and imaging. So the defective sperms can be taken away, and these magnetic properties add additional functionality to we can actually manipulate and sort the proper sperm molecule. The sperm can be isolated, so which is the having some damage or having good quality can be separated by using these luciferase magnetic nanoparticle composite technology. See, as I'm giving you the example, so here this particular the biomarker based nanotechnology for the improvement of these beef and dairy cattle is possible because I'm just sharing one example, one experiment wherein the lectin PNA, which is actually when there is a defecting in the sperm, there is a, you can see that there is a complete coloration of the sperm, and the sperm which is actually having intact and PWP is there properly, this protein binding protein is there, so which will show only the red coloration of the head, and the rest of the sperm tail which is not defective, which will not take any green color. So one can say that this the sperm which is having the red headed sperm without a green tail coloration is considered as a best sperm, so that can be isolated. So like based upon this particular technology, one can able to take the proper sperm for the conservation of the performance of the beef or the dairy cattle performance. And then the another very important area where the one can actually purify the nano purification of the semen. So the magnet used for the attract the nanoparticle coated defective spermatozoa. Those who are the spermatozoa which are defective, they can be captured using the nanoparticle base, and then one can discard the pellet which is having defective spermatozoa, sperm fragments and nanoparticles. And then the which are the very superior sperm which comes in the supernatant that can be collected and used for the further processing. Then there is another technology where the zinc oxide nanoparticles has been utilized for cryoprotection. This is very important area. Recently the people are actually trying to preserve the sperms of the good quality. So you can see that this particular experiment wherein normally what happens in a spermatozoa when they cryopreserve, there are fair chances of lipid peroxidation and membrane damage, and then there is a redox potential increases and the DNA get damaged, sperm DNA get damaged. So when you use the zinc nanoxide then zinc oxide nanoparticles, we can actually coat it onto the complete sperm, which resulted into the membrane stabilization. There is no formation of oxidative radicals, and which actually protect the sperm DNA with the sperm with the intact DNA. And this particular technology is really helpful. And then even though the many freeze thaw cycles we stand the freeze thaw cycles by protecting this one, because every freeze and thaw there is a fair chances of DNA damage, which is avoided by using this zinc nanoparticle as a cryoprotectant to store the sperms. Then there another very important area where people have used the nano water for ram's human cryopreservation, especially the rams where it's very difficult to preserve the ram semen. So this particular technology, the nano water, which is a water clusterized in the low temperature plasma reactor, which is this is the nano water. So one can use this nano water to preserve the insemination with the nano water, and resulted into 73% more good quality of sperms which as compared to the distilled water semen, the sperms stored in distilled water. So, and then the full term pregnancy for nano water has resulted more than 70%, and as compared to the distilled water stored ram semen. So this particular type of technologies, that biotechnological tools, then how one can make the nano water using the low clustered or the declusterized low temperature plasma reactor technology has been used to make the nano water, and this nano water can be used to preserve the sperms, especially for the endangered species or maybe the and then this particular most of the times the lamb preservation also very important, especially for the good quality breed. Then another one important technique, especially for the females, the ovum pickup. So this is a very normal technique, but which has really paved the way. This is a non-invasive, repeatable techniques which can be recovered by using large number of component oocytes from the antral follicles of the live animals, and then repeated ovum pickup can be done performed without side effects of both cattle and the buffaloes, with minimum stress to the animal. And as it's proud to say that the first time with Prasad and co-workers group at NDRI, they produced this Saubhagya, the first buffalo calf, using this particular ovum pickup techniques. And this has given a lot of advantages to collect the oocyte from farm animals with less invasiveness, and the superior animals can be used. And this oocyte can be used as a the donors in the embryo transfer method technology. And this is really important area where which has paved the production of the livestock and production, or we can say the embryo transfer technology. And then the another very important area which has paved the way, that is intracytoplasmic sperm injections. The when this successful, especially this particular technology was resulted in from the small animals, now it has transferred to the large animal as well. The success has been done in rabbits, mice, sheep, humans, and horses. And this technique is useful for the sperm vector system for animal transgenics. So when you are making the transgenic animals using this particular technology, one can use it. And this ICSI has been done with the sex semen, and more than 80% of the cattle are ruminants, and repeated freeze-thaw cycles of the sperms, which are after using the repeated sperm cycles, which is good semen, good sperm quality sperm can be utilized using the ICSI and straight away used for the zygote formation, or you can say fertilization. Then another very important area is the embryo transport technology, where it has helped to improve the livestock as faster rates, as well as the opportunity to genetic contribution to both male and female. And then the multiple ovulation and embryo transfer, which is established in early 80s. Late 80s, which has really helped to maintain the programs, resulting into the selection of intensity, and the reduced generation of intervals, resulting into the increased genetic ratio was possible because of the embryo transfer technology. And this particular technology is being used very regularly. So one can take the sperm taken from a bull from a very high-yield cattle or high-yield herd, and the cow is inseminated with that particular sperm. And when using the embryo split technology, one can divide the sperms into different, and then the surrogate females can get this embryo and then use it for the foster the mothers. Then the another very important area where which is not possible to, it was very difficult to store the embryos. So the using the vitrification technology, which has helped the concentration of the highly concentrated aqueous solution, including the glycerol, ethanol, glycol, and the non-permeating agents such like sucrose, glucose, or fructose. So vitrification is actually successfully used for the cryopreservation of the cattle embryos at very developmental stages is possible. And because of that, we could able to study many mitochondrial diseases or other related diseases which has actually transferred. So this has helped to develop the biotechnological tools also, and this is biology as well. So this is a very simple technique. So like slow freezing versus vitrification. So the vitrification usually takes place in the ultra cooling, rapid cooling method is in use as compared to the slow freezing. Then the very important area where the cloning. The cloning has, you can say, the first the dolly was cloned using the somatic cell nuclear transfer technology, and then after that the one can take the tissue biopsy from the animal donor cells in a grow in a culture, and then it can be actually where one can take this nucleus of the cell, the donor nucleus can be taken out, and then we can add the nucleus from the another cell type, and then we can clone and transfer it to the surrogate mother, and we can find the somatic cell nuclear transfer baby is here. So we can see it. So I'll just unravel these techniques in detail. What happens, this particular using this particular technology, especially in India, we could able to give the successful pregnancy for the clone cows like Garima 1, Garima 2, Mahima. These female were born with the this particular technology, and even the recently Sesta and Swarn was also produced. And then
याला ह्योच
espe-
तुम्हाला बोलला का पैसे देत आहे म्हणून.
and
all
then
the all the
you can see
sir,
how
sir,
I'll
one
just
minute.
explain you how exactly this particular technology, and by using this somatic cell nuclear transfer one can study the reprogramming events very well. So I'm just because when I was in US, we could able to derive the primate embryonic stem cells using this SCNT methodology, where we took the fibroblast same from the donor monkey, and then the oocyte from the oocyte, and then it was enucleated using the Splendor technology. So the DNA was taken away, the oocyte was taken away, and then added with the this fibroblast, and then the cloned embryo was actually formed here. The ICM was transferred, and then we could get the embryonic stem cells using the somatic cell transfer technology. So like this, we can derive the stem cells which can be utilized for specific cell type. So I'm just giving the example of fibroblast, but the cell types from any tissues can be taken, and then the individual specific somatic cell nuclear transfer technology can be utilized. So here I'll just give the how exactly we did it. We actually did using the ultrasound technology. We aspirated the follicles, and then the follicles were taken from these then the oocytes were isolated by using the Stalfeps technology, and then the oocytes were stripped, cumulus cells were taken away, and then to put it into the HCM9 medium for some time. So we get the mature oocyte. Once this mature oocyte is there, then they using the spindle technology, we taken away the histaining. We took away the polar body, and then the polar body was added with the another the fibroblast, and you can see these how the exactly the blastocyst development took place. The zygote, then 2 cell, then 4 cell, then 8 cell, then morula and compact morula. And once you this blastocyst, we taken away this inner cell mass from here, and this inner cell mass was taken away, and then put it into the feeder layer. So this program, so we took the immunosurgery using the rapid antigen technology. So we added these particular rabbit antiserum serum was used to separate the inner cell mass, and this inner cell mass was isolated using these things, and then manually passage for 5 to 7 days. And then to characterize whether this particular embryonic cells has all the characteristics. So accordingly, using this technology, we use these stem cell markers, NANOX, OX2, TGF, and left and third were used to confirm that these cells are of embryonic origin or having a totipotency or a pluripotency. So then they were separate. They were also characterized by using these markers. Then for further germ cell specific markers also for this particular embryonic stem cell, the somatic cell transfer cells were utilized using the embryoid bodies, and these different markers like OCT4, WASA, GDF2 were characterized. And then we also try to look into the immunohistochemistry whether WASA and OCT4 are there. And then we also try to whether these particular stem cells as SCNT derived lines are forming the teratomas. So accordingly, the teratoma was formed. So we confirm that this particular stem cell, so somatic cell nuclear transfer stem cells has these they can be used, and they can form the different teratomas into different lineages, and can go rise to the specific organs. And then we further characterize by cytogenetic techniques. Why I'm sharing these things, once you unravel this particular journey, so one has to use these technologies or these techniques to characterize the somatic cells or any technology which is being used. So accordingly, we compared these cells by doing karyotyping. They found the well characterized G banding pattern, which is normal euploid chromosome complement of 42 chromosomes like this. And we further did the microsatellite analysis, where one can see whether the females are there. So all these STR markers are intact, and they could able to see it. So the donor cell was completely transferred to here. So accordingly, one can get the case 3 and case 4 from the donor means there was no transmission from the oocyte donor. So the whatever the cell was derived only from the fibroblast. Accordingly, one can confirm that the whatever we enucleated from in terms of the fibroblast was resulted into the somatic cell nuclear transfer cell line. And then further, we look for the mitochondrial DNA sequencing analysis, and we observed that this is from the same female donor. Then we differentiated into different lineages to characterize whether they form the neurons or something like that. So we could able to see it, and the beating the myocytes were also seen, so that it also giving resulted into the cardiocyte formation. Then another one very important area which is actually being used for the livestock productions recent time, that is handmade cloning with chemically assisted enucleation. So where people are using the handmade technology, handmade cloning is done. This is the way one can actually do the handmade cloning, and then you can get the different hippers or the calves can be formed. And this handmade coding has a really very inexpensive methodology, less expensive required to the manipulation. There is no need of manipulation using the microscope. A simple, rapid, easy to learn and perform. And this particular handmade technology training is being offered at CIRB as well as the LDRI Karnal. So if those are interested, one can actually go and take the access of this particular technology there, and which is actually microfluidic technology also resulted into the formation of the specific sperm can be utilized. How exactly the sperm zygote to take place and can be studied using this particular technology. Then the in vitro maturation, fertilization, and culture is also one of the very important area. As I covering all this one, as in the first slide I told you the OM pickup, then in vitro manipulation, in vitro fertilization, then in vitro culture, and then embryo transfer. So one can take this particular all set one together, and then which is help analyzing the different potential of embryos, then pattern of gene expression, whether they have the intact pattern. So using these all these techniques, one can think about transgenesis, which we'll cover in the next slides. Then one can also study whether there is any impact of the environment, the epigenetic modification. Apart from the DNA, is there any impact of the environmental DNA onto the system? So epigenetic modification can be studied by using this technology. And then the cytogenetic disorders of the domestic species can be studied by employing these techniques in detail, so that we have a control of what we are transferring, what we are growing, and accordingly the specific animal model can be studied with the specific cell types, and then accordingly this technology can be utilized for further unraveling of the biology or the disease biology. And then coming to, as I told the last slide, that the transgenic animals. So see, all this technology has resulted paving the way though for the human mankind also. So using the recombinant DNA technology for the specific hormone production or the antibody production, for protein production, for diagnostics tools, gene function research, and then this has resulted in the increased growth rate. One can actually manipulate one gene, or there's like a somatotropin growth factor or the transmodine genes or modulation genes, one can actually do it. Then sometimes we can also look for the disease resistance. So one can make the manipulation using the transgenic technology, disease resistance can be done. Then the improve the nutrition quality of the food. So the meat quality can be improved by using this technology. Then the muscle mass can be increased by using the high fiber content, and the tenderness also can be improved. Then using this transgenic technology, better wool production also possible, and which has actually paved the way. Next subsequent slide will put some light on this one. And then the study diseases better, so one can actually understand the disease biology very well. And then the like biofarming, this is the whole thing called the biofarming, the transgenic animal advances in medicine and the research. So these are, as I sharing this particular slide, because the milestones and transgenesis in farm animals. It started with the early 80s, and then the first transgenic sheep pigs using the micro injection technology, and then coming to the transfusion transgenesis in pigs, and then so on, like these particular things. So these transgenesis milestones resulted into the formation of knockout pigs, and then gene specific pigs, and then even the SCNT technology also paved the way for random integration of the construct, which has resulted into the disease biology analysis. So as I told you, these are the different areas how exactly one can do the transgenic animals. The DNA transfer via direct micro injection is one of the very important area. DNA micro injections. DNA transfer via transposomes is one of the very important tool. Then a DNA transfer via lentiviral vectors, DNA transfer via sperms, which is actually sexed, or you can say isolated by different technology. DNA transfer via pluripotent stem cells, like as I told you, the stem cells can be used, and DNA transfer via cloning. These are the different ways one can actually do the transgenic animals. So as I told you, the embryonic stem method, which was explained in detail in the when I explained the SCM technology, where the embryonic stem cell with the desired DNA can be injected, the injection with the blastocyst, injection with the foster mother, then formation of new individual, and then the test observing for the presence of gene, one can do it, and accordingly this methodology can be utilized. So these are the different examples of transgenic animals. We go one by one. So the first one is Tanzanic cows, which was this particular Tanzanic cows carrying extra copies of two types of casein genes, which is giving more protein milk, proteinous milk. 30% more protein is there in this particular cow milk. So using the transgenesis, like this transgenic myostatin knockout cows has been produced in the US by using transgenic technology. And then this transgenic cow lacking the beta lactoglobin. Some people, some human beings, or some they're having the lacto tolerance, lactogen tolerance is there. So lactoglobin, so one can actually divide the lactoglobulin from this particular milk, and so one can develop this such type of milk also. And then the this omega 6, the having the healthier omega 6 or omega 3 ratio is also possible by doing the omega 3 fatty acid desaturated genome into the pig and cow, and accordingly this technology has resulted into the creation of the meat which is rich in omega 6 and omega 3 fatty acids, which is also a big gift from the transgenic cow to the human health as well as the nutrition point of view. Then the transgenic pigs, the environmental pig, this is case so enviro pig. This is fertilized by using normal egg with the sperm that has the incorporated foreign DNA, and by using the sperm DNA mediated gene transfer. Which has these transgenic pigs express the phytase in their salivary glands, and the phytic acid in the pig meal is degraded, releasing the phosphorus. This phosphorus is absorbed by the pigs, and normally the phytic acid or phosphorus complex passes through these pigs and excreted as a waste. And this pig waste is major pollutant and cause lot of problems. So the enviro pig excrete 30 to 70% less phytase, so which has resulted into the as compared to the conventional pig. So like this enviro pig is when there, so the phosphorus which was excreted from the as in terms of phytic acid in the into the pig excreta was really a nuisance. It was causing a lot of problems. So now because of this transgenic technology, we could able to develop the enviro pig, which has having less 70 to 30% less excreta, which is having less phytases as compared to the conventional pig. Then the transgenic sheep and goats, the actually when the first dolly, the Tracy, the sheep, the first transgenic animal produced with the recombinant protein drug for her milk. The milk was very important criteria. So wherein the alpha 1 antitrypsin treatment with the emphysema and cystic fibrosis was also considered a very important area, especially for the disease biology. So the transgenic sheep was developed, which was resistance for these particular things. And then they also created by the PPL Therapeutics and Rosen Institute this particular technology. So then the Tanzanic sheep and goat. See, the spider silk was regarded as the strongest material known. So using this transgenic technology, the genes from the drag-like silk were isolated and put into the goat using the transgenic technology in order to express the mammary gland from the form of milk. So accordingly, the milk giving this particular silk protein found in the milk can be used as a make lighter and stronger bulletproof waste or thinner threads of the surgery or cloning. So this particular spider silk was able. This was a gift from the sheep transgenesis technology, sheep and goat transgenic technology, wherein we could able to clone the drag-like silk gene from this particular species, and then we could get the milk having this particular silk protein. And this particular silk protein is being utilized to make the bulletproof jackets, and even for this particular the thread also for the surgery, especially the inner organ or internal medicine surgeries or various fine surgeries. This particular silk is being utilized as a toughest molecule so that it can sustain for more injury or something like that. Then the Tanzanic chickens. This is also okay. As why Tanzanic chickens? One can have a very important question because they grow fast than the goats. So there are several methods, especially the infecting embryo with the viral vector carrying the human genome. So then also the Tanzanic chicks, or this now these days another and I don't have the slide, but I can share one very important area where people have actually using the egg antibodies, like Immunogenics, one of the company in Hyderabad, who they are actually producing the antibodies using the egg yolk. So you can see the every time when you have to bleed the animal, then isolate the proteins. But we inject the antigen into the animal or into the hen, and then we can get the egg, and the specific antibody can be isolated. So IgY antibody, which is from poultry, is also the less cross-reactive with the other species can be get, and you get a lot of yield also. And this particular technology has really paved the production of more antibodies from the egg animals. So like the Tanzanic chicks, also one very important area which is giving us the more proteinaceous eggs, which is also help for the nutrition point of view. Then these human pharmaceuticals produced in the milk in the transgenic animals. You can see these are from the cow lactoferrin, using the cDNA as a transgene source. So one can get the using the promoter of alpha S1 casein. So you get the expressed milk, which is lactoferrin rich. Then the human lactoferrin, lack of human lactoferrin also been possible. Goat antithrombin through, then pig factor 7, rabbit calcitonin, then the erythropoietin proteins can be formed. And these are the different, this is the gift from the transgenic animals to the human pharmaceuticals, and which is being regularly utilized for the human mankind as well as the nutrition point of view. Then the efficiency of the producing transgenic animals. So using the different technologies, as I told you, the micro injections, nuclear transfers, and the retroviral injections, then the producing the historical, like this technology has really paved way to produce the transgenic animals of specific cell types or specific characters, and which has resulted into the livestock production enhancement. Then the conservation and cloning, very important area, because these days we are facing the problem of indigestion. So the extinction is going on for most of the species. So mammoth, then kaga, picardo, and gore, these are the very important species being considered very importantly. So conservation cloning is being adopted for actually preserving these important species. Then the biotechnology analysis, and then selective breeding also possible, application in the genetic improvement. So another very important area wherein without any technology one can use the genetic or genetic background knowledge. You can use the genetic improvement by find easily detectable genetic differences among the animals, and then develop the specific SNPs panels so for to detect those differences, which is possible. So accordingly, like human being also, SNP is being utilized. For animals also, one can use this technology, develop the SNP panel of the specific differences, then they relate these SNPs between the productivity differences. So though high producing cattle versus low producing cattle, or high producing the good quality milk, or maybe the even the percentage of the calving also can be considered, and then accordingly the SNP panel of 1 versus 2 can be compared, and then the productive differences can be analyzed, and then the rank the according, and these rank these animals according to their economic merit. And these the for, and then these parents can be utilized for next generation for the selective breeding panel. So the for there are a lot of avenues are available, especially for conservation as well as for the genomics part, especially the federal support for US government, as well as a lot of federal FDA also giving a lot of support for genomics and to analyze the genomics, especially for cattle, sheep, swine, poultry, and horses. And even these days the aquaculture, fishes and other animals also possible using getting funding to preserve or support is available from the federal governments. Then how one can use these genomics? As I told you, one can identify the DNA sequences associated with any disease resistance and the production trait, and then they can be evaluated based upon their DNA from the birth as well. Then best animals can be parents can be determined earlier or more accurately using this genomics technology. And then very important area, this is recently this particular area is being considered traditional animal breeding method, that is TABM is there, which is slow. And then the progeny testing for production test takes 3 to 4 years for insemination, but bull will be at least 5 years older before first evaluation is available with using this particular technology. But now one hand, but these particular things can be considered very positively, because progeny testing costs around say 25 to 50,000 US dollars per bull, and then only 1 to 8 or maybe 10 bulls graduate from the progeny testing. So after analyzing all this data, one can get maybe one or two. So this particular investment is more than $2 billion. So using this to avoid this type of such type of technology, when there is a marker assisted selection, mass selection technology is being utilized. So where is the segment of DNA at a unique physical locations of the genome that varies sufficiently between individuals with their inheritance can be tracked through the families. So using the marker assisted selection technology, one can track one specific gene, and then which is markers are not required for part of the full gene. So only small area can be considered. And then genomes is this as I told you. So genomics for each individual is available. One can make the selection, and accordingly the genes can be tracked. So these which really help to allow the inheritance to be followed in region across generation. So one can actually from the generation to generation a specific area, one can follow the generations very tightly. And then the SNP are the markers of choice. So accordingly, as I told you in the last slides also, so you can make the SNP panel of this particular cell types or maybe particular species, and then they can be used as a marker. So there is no extravagant technology like STR markers. Only these small SNPs can be considered, and then only need lots. So 3 millions of genome is possible. So see, one can have these genomes are available worldwide. So accordingly, the SNP can be made, and then their specific subsequent data is also available for analysis purpose. So see now, so considering that these are the different companies come forward and develop the SNPs, single non-nucleotide polymorphism also been developed for the animal species. So these are the different sources which are available. So one can make use of this best of the use of these SNP panels which are available for the different animal species. And then these reproduction and technologies have potential to combine the adopted traits for human local breeds with allied breeds to raise milk and milk productions. As I told you, this is the giving us the area, because we are actually again going back to the preserving our their ancient breeds also, which are having the disease resistance as well as having good characters, A1, A2 milk or characters also there. So this recombinant DNA technology along with the traditional trait of approach can be utilized to preserve the to create the good quality of milk as well as meat production. And then this as I told you, this particular program, the Brazil is the one country which has actually taken most pride there. Holstein cow, JP Gir cow. So these are the heat and trophic resistance insemination was done, and then they got a good breed which has a crossbred and has showing me more than 80% of the total milk in the Brazil, which is produced from this Giralando cow. Then the IVF or in vitro fertilization also paved a lot of way, especially in the Brazilian technology. Why I'm sharing this is because they are the one who have pioneered this particular area of ETT as well as in vitro fertilization, and they preserved the specific things. So you can find out our own desi cows or buffalo these traits, or we can say semen these characteristics also with them. So because they preserved this particular using this technology, they preserved these particular zygotes or you can say characters with them. Then the genomic selection, you can actually skip this one, which I already covered earlier. And then, but of course, there is now because of the biosafety things and all those subs, we can see these lot of trans variation also coming up. So then we have to coin this, utilize these techniques with risk and regulations are there. There is a concern that the food product derived from these transgenics or cloned animals may pose human risk to the human health as well as the animal health. Also, there could be a transformation, there could be antigenic drift also takes place. And then there is also concern about the potential impact of the biotechnology on the environment and animal welfare. Yes, that is there is a risk, but we have to accept this risk and then judiciously using all the biosafety norms and making all the records possibly we can regulate this structure adequately and to evaluate the control risk of these particular technologies. And then this is very important area. Then, as I told you, the consideration and concerns also there. For especially the human health concerns, transplantations may offer the benefits. There is some risk of including infection rejections. Then the food safety concerns. So potentially food safety concern about the product derived from the genetically engineered animals also has some problems. And this we have to coin. This both the technologies are definitely paving way forward. They are always beneficial, but one has to utilize this risk, or the potential risk of generation of these pathogen free animals or recombination vectors can be possible, and one has to actually make the adequate safety norms following this particular technology very judiciously. All record has to be maintained, excreta or whatever discard is there, that has to be discarded. That's why the biosafety committee, especially the RCGM and other committees, has come into picture, which are controlling, regulating such type of research as well. So animal health and welfare concern, the ethical discussion, as I told you, this is very important area. Then environmental consideration, which is also been taken, especially the aqua advantage, salmon, which was very important area, which was considered in USA, which has, when they increase the salmon production, that particular area, other species were not growing. So which has really, the normal salmon stock was actually paving the way there, because salmon is very good for food, but particular area is devoid of that particular, any other species is growing. So accordingly, environmental concerns are there when using the following the any DNA technology or recombinant technology, biotechnological tools. One has to use them very judiciously. So to conclude, these technologies is based upon the modern biology, offers enormous potential for this livestock production. Preventive measures like vaccines, medical products, and other veterinary products also possible because of the gift of the biotechnology. However, these reproductive technologies, emerging techniques should be judiciously supplemented with good practices of animal health. Nutrition also need to be taken care. Management of all the level of stakeholders like manipulation, improvement of the health, production, and reproductive performance of the any livestock species need to be considered very judiciously, which will definitely facilitate the production and dissemination of the superior germplasm, thereby enhancing the overall productivity of the livestock species, which is possible. Everything is, the science is flared up. We can use these particular technologies very well, but definitely we have to use the cautious approach as well. So it is necessary to speed up the establishment of a comprehensive and effective framework for the safe use of these technologies from the dual standpoint of view, biosafety and regulations. Thank you for your patience hearing. I'm happy to take some questions as well.
Yeah. Sure. So very nicely, Dr. Vikas Dige has been presented regarding the comprehensive aspect of all the species of these livestock to enhance their production and different recent whatever those technologies are there for the purpose. He has been elaborated very nicely, and I think all the participants have enjoyed and got benefited with this. In spite of that, if somebody has some doubts or questions, they can ask, please.
Hello, I am Dr. Kembavi. Any control over this is there, or any supervision or government control over this is there of
Yes, sir.
cloning
Yes,
and all
sir.
that?
Yes, sir.
Yeah.
Actually, so now it is mandatory. The biosafety, the DBT has a biosafety committee. So each institution has to have a biosafety committee, which is monitoring the DNA manipulation, recombinant DNA manipulation, as well as now these days the even recombination of the bacteria virus has also been manipulated very the monitored very keenly from the biosafety committee. So there is a good control, and without any biosafety approval, one cannot undertake such experimentation for the livestock production as well as for the vaccine production.
Thank you, sir. Thank you very much.
Yeah.
Any more questions, please, participants? I think Dr. Vikas is explained very nicely, so there are no many questions. So now I request the next speaker to please join. Now the next speaker is Dr. Devaraja Naik. So whether Dr. Devaraja sir is joined, please confirm.
Yes, madam. Yes, madam. I'm here.
Yeah. So to just give a brief information in biodata regarding Dr. Devaraja Naik. So Dr. Devaraja Naik is presently working as a professor, Department of Livestock Product Technology in Veterinary College, Hassan. He has completed his BTech degree from Dairy Science College, Bangalore. He has completed his MSc in Dairy Science from Bangalore only, and he has completed his PhD degree in dairy chemistry in 2018 from Dairy Science College, Koksu, Bangalore. He has vast work experience in the field of academics, as including the industrial field. So total 21 years experience he has. As far as this industrial experience is concerned, Dr. Devaraja Naik is worked as assistant manager in Nandi Dairy, Nandiya, Andhra Pradesh. He has also worked as a plant manager in Sri Siddhartha Dairy, Madanpalli, Andhra Pradesh. And he has also worked as a deputy manager in the National Dairy Development Board, Anand, Gujarat. As far as academic experience is concerned, he has been worked as an assistant and associate professor in Karnataka Veterinary and Fishery Sciences University, Bidar, from 2006 to 2018. And presently, he is working from 2018 as a professor and head Department of Livestock Product Technology, Veterinary College, Koksu, Hassan. As far as his achievements are concerned, he has been conducted many trainings, like training programs on milk quality and clean milk production practices, installation of automatic can lead opener, installation of bulk milk coolers, installation of automatic milk collection, etc. So he has been very crucial in conducting very good type of the trainings in the field of industry. In teaching, he is actually actively involved in undergraduate teaching of BVSC and AH students, and also he teaches different courses of dairy technology. He has been guided PG students as a member of the research committee in four PG students, as well as he has been handled as a principal investigator the research 5 research project, and there is one more research project which is going on. He has been handled 4 research projects as a co-principal investigator. He has been also involved actively in the extension works, like conducting trainings, workshops, and delivering the talk as a resource person. He has 17 research article publications, and he has also published 5 manuals and 5 books. So with this biodata, we are very fortunate to have Dr. Devaraja Naik to deliver today's talk on present and future scenario of quality of milk in India. So Dr. Devaraja, please.
Thank you, madam. Thank you. Is it visible to all? Is my slide?
Yes, sir. Yes, sir. Just make it on presentation mode.
Thank you, sir.
I think Dr. Vikas Dige, I forgot to thank you. I'm very much thankful for taking out the time and delivering a very nice lecture. Thank you so much, Dr. Vikas Dige.
Thank you, madam.
It's sent already.
First, I would like to thank the chief patron of Professor A.M. Patarkar sir, the Honorable Vice Chancellor of AFSU, and also Dr. R.C. Agrawal sir, and the patrons, and also the course director, Professor A.S. Ranade sir, and the another course director, Dr. R.K. Zende sir, and also the organizing secretary, Dr. Villas Vaidya and his team, the co-organizing secretaries, program coordinator, co-coordinators. So, and the speakers yesterday, Dr. M.L. Madan Saab, the Padma Shri awardee, and Dr. C.K. Tota Saab, and also today's speaker, Dr. Vikas Saab. So, very good morning to all. And I thank to the organizer for giving me an opportunity to speak about the present and future scenario of milk quality in India. So the presentation of mine, it will flows in this way. Or the present global scenario of dairy industry, the overview of Indian dairy industries, milk supply chain of Indian dairy industry, present status of milk quality in India, production of dairy and value-added products, milk safety regulation and health management, key challenges, future prospect of Indian dairy industry, and then SWOT analysis. There are certain success stories also been explained, and the final part is the conclusion. See, India is a leading partner in the milk production, which is contributing about 22% to the global dairy production. So world production is about 937 million metric ton, which is increasing about 1% as compared to the earlier last year milk production. But compared to the last year, there was compared to the last 10 years, from 767 million milk to it was rose to 937 million tons during 2022. There are certain countries which are producing high surplus milk, like New Zealand, USA, France, Germany, Australia, and Ireland. So India is producing around about 189 million metric tons annually. There are certain countries have a deficit in milk productions, like China, Italy, Russian Federation, Mexico, Algeria, and Indonesia. Among the global, around 130 million households are engaged in the milk household business. So talking about the consumption of the milk globally, India is consuming 160,000 million tons, followed by the European Union and USA and China. So the highest consumption of the market lies in India, and then USA and European Union and China. But the per capita consumption countries, among the top 10 countries, Finland, Sweden, Netherlands, Switzerland, all these countries have the 361 gram per day per capita consumptions. So world largest dairy exporting players, even though they are producing very less amount of milk production, but their export is Germany is producing 18%, New Zealand is also contributing same of 18%, France, Netherlands. These are the major players in the international dairy export of dairy products. But when we are talking about the Indian dairy scenario, the GDP of the agriculture, it is contributing about 33,94,000 crores during 2020, and the livestock is about 9,682 crores. Among these, 18.4% is contributing from the agriculture to the GDP, and the livestock is about 5.2% contributing. India is the first in a bovine population, which is about 299.6 million, which is the average milk yielding of cattle is about 1,77,000 kg as compared to the global, which is giving very high of about 2,699 kgs. So India, the cattle population, the production of milk is less as compared to the global average yield. Milk production has been growing at the compound annual growth rate of 6.43 as compared to the world annual growth rate is about 1.53%. But in India, the per capita availability is 429 grams per day, which is above the ICMR recommendation, which is about 280 grams per day. So the consumption rate, it was 84 kg per year. Therefore, there was a huge surplus milk available in India, which can be divided into the milk which is collected in the organic sector is about only the 40%, and unorganized sector is about 60%. So talking about the roadmap of Indian dairy, so during 1950 to 1960, India has felt the deficiency in milk production. During 1965, the government of India has set up a board of National Dairy Development Board, which is taking care of the replication of Anand model or the Amul model throughout the country. So under this, the chairmanship of Dr. Varghese Kurian, Operation Flood program was initiated in phase 1, phase 2 and phase 3 during 1980 to 1996. So that right now 14.11 million farmers are depending on the dairy sector, in which 1,33,349 dairy cooperative societies are involved, 177 milk unions throughout the country, and 15 federations are involved for restricting the dairy industry in India. See, comparing the 2007 and the 2019-20, there was a drastic exponential increase in the milk production. See, during the 2007, there was 100 million tons. Now in 2020 and 22, it was rose to about 198 million metric tons. India is also exporting the dairy products, which is about 1,08,711 million tons, which is worth about 290,000 crores during 21 and 22. The major exports of Indian products to the neighboring countries like Bangladesh, the UAE, then Malaysia, Saudi Arabia and Qatar. But talking about the Indian milk production, that there are major states which is contributing for the Indian milk productions. The UP, which is contributing about 14.9%, Rajasthan is about 14.6%, Madhya Pradesh, which is producing 18 million tons, and then Gujarat and also the Andhra Pradesh. So these are the major states which is contributing for the milk production in India. The consumption and productions, as we compare to the Indian scenario of Indian dairy industry, see, during 2010, it was about 82 million tons. As compared to the production, it was goes to 203 million tons. And similarly, during 2016, there was 105 million tons, and the production was 10.49 million tons. So, from this data, we can come for the conclusion that the production rate is high as compared to the consumption. So that there is a vast surplus of milk which can be converted into the different products. This is about the international market of Indian dairy industry. India dairy product exports exclusively the milk and cream concentrated as sweet products, which is about 89% as compared to the milk, cream, not concentrated products. So, but whereas in case of the products which has been exporting to the neighboring countries, the main is the Bangladesh, Algeria, Saudi Arabia, UAE and Egypt. So these are the countries which we are exporting the products to the neighboring country. In that skimmed milk powder, which is about 220,000 million tons, almost like 11% is being exported to the neighboring countries. India's products which is being imported from the other countries, in that France, New Zealand, USA and Netherlands. So these are the 4 major countries which has been importing the products to the Indian But the market of Indian market of dairy products has grown rapidly over the last few decades because the faster the growing rate due to the change of the consumption and the mindset of the urban population is focusing mainly on the value-added products rather than the fluid drinking the fluid milk. So this is the recent change in the mindset of the people that they are slightly diverting towards the conscious about the quality rather than the quality. So this Indian dairy infrastructure, which is about the chilling facility from the cooperative sector, it is having about 464 cooperative societies are there in India, and the producer companies are about 10, and the private players are 293 chilling facility in our India. So along with that, the processing facility in lakh liters per day, the cooperative is having 663 lakh liters per day, and the producer company is having 25 lakh liters per day, and the private players are having the facility of 733 lakh liters per day. So among this, India is having 254 milk unions and the 9 producer companies. So the total annual milk production of 188 million metric tons. It has been divided into 40%. It goes to the organized sectors like private dairies, cooperatives, and the producer companies. The rest of the 60% of milk is been utilized under the unorganized sectors like the local vendors for their self consumptions. But whereas it is of 40% of the milk which has been collected through the organized sectors, that the 60% of milk directly goes for the liquid milk consumptions. The rest of the products, like 30%, is used for the different products, the value-added products. So most of the milk is used for the used as a liquid milk in our Indian dairy. There are certain products which have been available in our Indian market, like fluid milk, either in the bulk or in the toned milk or double toned milk, standardized milk, full cream milk, and UHT milk. But fat dairy products are cream, butter, ghee, and etc. Whereas the fermented products are in the yogurt, curd, lassi, and coagulated products are cheese, paneer, and channa. Whereas the dried products are condensed milk, milk powder, and infant milk powders. There are certain value-added products are available in our Indian market are lassi, dahi, fresh paneer, then buttermilk, cheese slice, misti dahi, milkshakes, cold cake, cold coffee, and then mango Greek yogurt, flavored milkshakes, chocobar, then flavored milk, dairy oatmeal, and lactose-free milk. So these are the different varieties of milk and milk products are available in our Indian dairy market. Apart from that, India is also utilized the 40% of milk is used for the preparation of traditional dairy products. In that, the fat-rich dairy products like ghee, malai, makhan, and the heat desiccated products like paneer, channa. From channa, the different products like sandesh, rasagolla, and pandava. From cultural products, dahi, it will be either it will be lassi or shrikhand, and the misti dahi is another one product. Whereas heat desiccated products, the products like kulfi, rabri, basadi, kova. From again the kova, the base the base products is kova, which can be converted this product into peda, barfi, kalakan, and also the gulab jamun. So talking about the present scenario of milk quality in India, in Indian system, we are collecting the milk through can, through bulk milk coolers, and through the chilling centers. So while collecting the milk, in the can, the average acidity of milk is about 0.162% of lactic acid, which is higher, and the pH is also 6, and the MBRT is 1.3 hours. But whereas in case of this, the bacterial count, the standard plate count, which is about 15 lakhs to 18 lakhs. But the standard says that if the milk is very good, it should be always less than 2 lakhs of bacteria, the coliforming unit per ml of milk. But whereas in case of the can milk, the presently what we are receiving to the Indian territory on an average, it is lies between 15 to 18 lakhs. But and also the presence of coliform, that it shows the sanitary condition of the milk, what we are handling at the either at the farm level or at the chilling center level or at the dairy level. So whereas in case of the milk which has been received from the bulk milk cooler, these bulk milk coolers are installed at the village level, and the spontaneously the milk is being cooled to the less than 5 degrees centigrade. The acidity of the milk is lesser as compared to the can milk, that is about 0.142% of lactic acid, and the MDRT is very high. If the MDRT is very high, the milk is very good quality. Very good quality. And also the most important factor is the number of bacteria which is present in the milk, which is about 7 lakh to 8 lakh. So that means the milk is in the range of good quality. And the milk which is received through the chilling center, so that the acidity of milk is 0.153 and the pH is about 6.2, but whereas the immunity time is 2.5 hours, but it is not so much been the difference in the quality of milk is received to the can and the chilling center milk, which is about 13 to 15 lakh of the bacteria. Whereas in case of processed milk, it means the milk which has been pasteurized, the acidity is slightly 0.158, but the milk which has been received at the dairy level is about 15,000 to 20,000 of bacteria, 15,000 to 20,000 of bacteria in the processed milk. So talking about the quality of milk, what kind of milk should be? At the milk, it should be contains high fat and high protein. The present scenario of the milk, which is normally we are receiving at the cow milk, we are receiving is about 3.8 to 4.2% of fat and 4.2% of protein. But the milk, which is been best quality milk, which is having high fat, which is about 5 to 5.5% fat, and it is about 4 to 4.5% of protein. And also the milk should also contains low number of bacteria. So there should not be any foreign materials like dust, dirt or flies in the milk. And also it should be free from the contaminants like the antibiotics, pesticide residue and aflatoxin residue. And there should not be any alterations in the products. And the somatic cell count should be lesser. It should be always less than 3 lakh of CFU per ml. And also the milk should also be received from healthy animals. And the person who is handling the milk should also have the healthy sound. See, there are certain bacteria which are being beneficial and also the harmful. The bacteria like Lactococcus, Lactobacillus, Streptococcus, so prokaryotic bacteria and bifidobacteria, these are the bacteria which have been very useful, and it will be useful for the preparation of dahi, lassi, and yogurt and shikhan fermented products. But there are certain bacteria which are causing harm or spoilage or which may be cause a disease to the consumer. So those are the bacteria which may get through either through the air or water or through the feed or through the utensils. So certain bacteria are like Pseudomonas, the Clostridium, the Micrococcus, the Bacillus species, and Listeria monocytogenes, Salmonella species, and E. coli bacteria and Campylobacter. So these are the bacteria which causes the disease or to the illness to the consumer. The source of contamination of the organisms into the milk, one through the interior of the udder, exterior of the udder, and the skin surface which is been touched, animal sheds, the milking staff, the person who is handling the milk, and the milk which is utilizing the utensils, and also the quality of the water and also the air. So these are the parameters which will contribute to the contamination of the microorganisms to the sterilized milk. The milk which is been comes out from the udder of a healthy animal, it's almost like a sterile condition, which is about 200 to 500 colonies or bacteria which are there in the milk, which is the milk which is comes out from the healthy animal. Once the milk is been comes out, then it starts propagating, and once it is reached to the dairy plant at the ending stage of processing, it is about 15 to 20 lakhs of the bacteria. So this is how our Indian condition that the milk will get contaminated during the transportation at the stage of the village level, or at the dairy cooperative level, or at the chilling center, and the transportation, the mode of transportation, and at the dock where the milk is receiving for further processing. So all these parameters are encountered for the increasing the bacteria in the milk. So microbial contamination takes place from the different sources of. So during this bacteria count, the fresh dung, which is contributed about 1 lakh to 634 millions of bacteria is contributed, and also the mastitis and the subclinical mastitis, which is about 1,000 to 3,000 millions of the bacteria are contributed, whereas the 4 milk from the teat canal, which is contributed about 600 total bacterial count as compared to the middle milk and the strippings, which is drawing at the end of the milking. So the strippings contains 400 of bacteria as compared to the four milk, which is contributed about 600 of total bacterial count in the milk. And also the wet milking is one which is contributed about 1 lakh of bacteria. And also the flies, which is about 11 lakh 22,000 of bacteria is contributed, and it is get contaminated in the milk. And also the utensils which has been uncleaned is about 25 lakhs, and washed or steamed utensils are contributed about 25,000 of the bacteria. So this is how the bacteria are one or either way these kind of bacteria will get contaminated with the pure milk, and the bacteria will get multiplied. So solution is that they make the milk aspersis. So by using the different heat treatments like pasteurization, heat the milk to the pasteurization temperature of 72 degrees centigrade for 15 seconds, and sterilize, or you can go for sterilizations of about 121 degrees centigrade for 15 seconds. And also we can reduce the bacteria by use of low temperature like freeze drying, and also for the drying of milk. And also we can adapt the refrigeration storage of bulk raw milk at the farm level itself, so that we can retard the growth of microorganisms at the farm level itself. And also delivering of these products. So the marketing of products, the processed products in either in the insulated tankers or the trucks is a most important parameters. So another important thing is the adulteration of milk. So milk, nowadays 65% of milk what we are consuming may be adulterated. So the common adulterants are the urea, the water, starch, sugar, and also certain additives. Along with that, there are certain preservatives like formaldehyde, hydrogen peroxide are also been abused to the fresh milk, so that these formaldehyde is act as a preservative to enhance their milk. So the main cause is the cancer, skin rashes, or eye diseases. Whereas in case of the hydrogen peroxide, it may lead to the cardiological problem. The use of urea, which is having high amount of nitrogen content, so this will lead to the high protein content in the milk. It may lead to the failure of kidney, heart, and liver. And also there are certain vanaspatis or margarine is also been used to to the ghee or butter to increase the fat level. The starch, what they are using to increase the thickness or viscosity of the products so that it can lead the obesity to the consumer. There are certain additives also being used in our Indian milk to increase to add as a flavor, the coloring agents, the sweeteners, which will act as an antioxidants or antimicrobial to the product. So the quality issue is talking about the food safety is a primary concern to the regulating bodies. The FAO or WHO or FSSAI, all these regulatory bodies are the main concern is to make the food safety to the consumer, so that to ensure the food is safe and nutritionally secure. For that, milk it should be free from the chemical and the microbial quality, so that the chemical contaminants are one which is being antibiotics residues, the pesticide residues, and the heavy metals. Along with that, there are certain allergens, as I discussed, the preservatives and also the toxins. So this is how the milk will get contaminated through the agricultural or veterinary practices, use of antibiotics, the hormones and also the agrochemicals. So through the environment, either through the air or feed, water or soil. So especially the mycotoxins in that the feed, especially the milk, it is been the mycotoxin is aflatoxin M1 and agrochemicals and metals. So there are heavy metals are also they get contaminated in the milk. So through the milk utensil, the metals are additives during the heat treatment with animals. During the cleaning process, the sanitizer and disinfectant, which is being used for the milk preservatives, are also get contaminated to the raw milk. So the metals in the final products, the heavy metals, the additives, the migrants are also get contaminated either during the processing or packaging. So these are the ways or pathways that the milk will get contaminated in different ways. The use of antibiotics to the animals for treating the animals to the diseases, that there are certain risks of antibiotics residues in milk. The consumer safety is one which is emerging the development of antibiotic residues or antimicrobial resistance to the consumer is one of the greatest threat to the human health is concerned. And the regulatory issues like national and international standards is also compliance to the standards. But the processing is, we can get the economic loss, but this processing is usually inhibits the lactic acid fermentation, especially with the preparation of the cheese, paneer, or any other kind of yogurt fermented products, and also it will develop it also hinders the development of flavors. Apart from that, the presence of antibiotics in the milk, which is responsible for the renal clotting time, so that renal clotting time will be prolonged, so that the improper clotting takes place. As a result of that, it is a huge affects the products like cheese, or paneer, or yogurt, so that the improper ripening will also takes place. Therefore, there is also undesirable growth of antibiotic resistance of starter culture bacteria. And also the phosphatase test, which has been the alkaline phosphate test, which has been used as an index for the pasteurization of milk. So there may be chances of giving false report. So these are the chances of in the milk processing difficulties concerned. Whereas in case of health issue is concerned, the FSA has recommended certain the tolerance limit that mononsin, sulfadamide, and then linomycin, neomycin, streptomycins. So each components of the veterinary and veterinary drugs or antibiotics, it has been recommended to the tolerance limit of 0.02 for menancin, and streptomycin, it is for 0.2 milligram per liter of milk. So if the tolerance limit is crossed and the animal is get contaminated or been administered with these kind of antibiotics to the animal, either to the food or feed or to the water or any other kind of source, then it will go to the animal products. Either it may be meat products or it may be dairy products. So these animal products, again, it will go to the human body where there is a physiological disorders, and also it may lead to the toxicity and hypersensitivity or hormonal disturbance will also be takes place. Along with that, it may lead to the cancer and immunosuppression and the mutation of DNA, then the tetragenicity. So there may be chances of the gastrointestinal disorder. So these are the many factors and also be affects in by the abuse of antibiotics residue or veterinary drugs to the animals over the above the tolerance limit to the final products of the products of dairy. So there is heavy metals as also recommended by FSSAI. If for lead it should be less than 0.2 ppm for infant food and infant substitutes. But whereas in case of the international standards, Codex Alimentary, it is recommended only the 0.02%, 0.2 ppm. And for ice cream, it is only the 1 ppm, whereas in other milk products like cheese or butter or other products, it is recommended for about 2.5 ppm. And the certain heavy metals like cadmium, mercury, arsenic, tin, zinc, copper. So these are the heavy metals which may be get contaminated either through the feed or water or through the any other source of modes. So these are very health affecting metals which may lead to the pancreatic disorders, leads to the cancers from this kind of heavy metals. Another one is the mycotoxins. These mycotoxins will grow as molds on the crops. They may produce toxic substances that can remain in the crops. So these are the mycotoxins which may get transferred to the milk is in the form of the aflatoxin ammonia. So there are certain 100 lives of outbreaks was happened in the USA, which may lead to the cancer and the immunosuppression and stunting of growth is also been takes place by consuming these kind of aflatoxin ammonia in the milk. There are certain hormones which may be used as a growth promoter or in the livestock for to treat the animals, but this will also a serious concern to the contamination either through the water drinking or fecal or urine excretion. So these hormones which has been administered to the animal like insulin or oxytocin are the hormones which has been get contaminated through the animals to the products. So these are the hormones which may cause breast cancer to the consumer, and also there are certain links has been made to the hormones which is having the residues, and it may lead to the early puberty in the girls. So FSAI has conducted a certain surveys during 2018 that certain samples were also shown the presence of aflatoxin ammonia in the ammonia residue in the chromosomal lipid, and also there are certain samples also presence of the antibiotic residues in the milk. So another is the cause of mastitis. This mastitis is caused by the inflammation of one or more quarters of the udder of the animal. So normally the inflamed udder, it shows the swelling and also kind of pain feeling, and it is was warm and looks like reddish in nature. So these inflammatory infections is caused by the many bacterial infections by like mycoplasmal or E. coli, the streptococcus and staphylococcus and corona bacteria. So these are the bacteria which may get entered through the teat canal and it will result into the mastitis infection. So there is a huge loss to the producer, which is accounted about the 26% of the milk is been reduced. The cost of the milk is reduced about 26%. And also the yield of the animals is also been, the yield of the milk is also been reduces. So, so such milk, this is mainly due to the reduced milk production, and such milk we have to be discarded, and early cow replacement cost has to be made, and reduce the cow sale value, and also we have to reduce the use of drugs, veterinary services, and also the labor. So these are the losses to the owner of the animal, which the animal is get infected. There are certain factors affecting the development of mastitis, one mainly by the unsanitary conditions, and also the improper use of the milk machine, which has been the improper use of the vacuum pressure, which may lead to the trauma to the animal, and also the anatomic and structural weakness of teat canals, and the entry of pathogenic bacteria through the to the teat canals. So these are the factors which may get the animals the mastitis infected. So this practices to avoid to obtain the best quality of milk, we have to adopt the good hygienic practices. Start from the village level, at the from the animal level itself, that the hygiene of the animal, the cleaning of the animal, and the shed, the area of milking, the personal hygiene of the milker, and also the environment and the equipments, the utensils, what they are using for the milk to carry out to the either to the dairy co society or to the milk producer company. So all levels, the hygienic should be maintained, and also to follow the good manufacturing or good managemental practices. So these, the managemental practices at either at the society level or the chilling center level or at the plant level. So all this area, so it has to be managed that should not get contaminated either by the cross contamination of the products or should not be get contaminated with the foreign materials. So along with that, the implementation of clean production practices start from the village level. It has to be completely managed by the implementation of HACCP at all the stages of milk, which has been enrooted to the finished products. So application of hazard analysis critical control point, which has been internationally recognized and accepted to take care of the milk or the products, either the agriculture products or the meat products or milk product, it has to be curb the contamination at the primary level itself. Therefore, this is a quality assurance of to the consumer rather than the quality control to the products. So this hazard analysis critical control point, which has been internationally accepted for food safety management system, so that it ensures that the food is safe and is safe for the consumption and it will not cause any harm to the consumer. So there are certain key challenges of the dairy industry is concerned, our Indian dairy industry is concerned. So India is producing low productivity of bovine animal. We need to enhance this productivity of the animal of the capacity which has been yielding to 500 to 800 kgs per lactation we need to enhance that of the animals to 1,500 to 2,000 liters per lactation. And also imbalance of feed into the animals. So limited access of milk producers to the organization sectors. So hardly India is collecting the milk from the agri sector at about 40%. So rest of the 60% goes to the either the local vendors or they will consume in at the their production level itself. So this it has to be increase the collection of the 40% to the make the organic sector to the 60% of milk collection to the organic sector. The age-old infrastructure operating is an obsolete technology. So during the Operation Flood program, we have made the doing the strengthening of the dairy plants. The old equipments, operating equipments are still existing. That it has to be make the machineries as an automatic system of processing, and also lack of manufacturing facility for the value-added products. So value-added products like the preparation of the lactose-free milk and the fortified milk of the vitamins and also the whey protein concentrates and the fat-free milk. So these are the value-added products. It has to be made the facility to make available in India. So and also lack of efficient chilling infrastructure at the village level. So government has put lot of investment to chill or to make the best quality of milk at the village level rather than going at the chilling center level or at the plant levels of taluka level or the district headquarters. So chill the milk at the village level so that the proliferation of bacteria should not take place and it will be the best quality of the milk will be obtained by chilling the milk at the village level itself. And also the lack of efficient chilling infrastructure at the village level and the awareness on the hygienic practices. So from where the contamination will take place, the use of the personal hygiene or the sheds, these are the area parameters has to be considered for the hygienic practices. And also the continuous trainers for the training programs has to be conducted to awareness about the quality of milk, the contamination, the pesticide residue, like contaminants, and all these things has to be accountable for the trainers to train them to get the awareness about the milk quality. And also lack of penetration in small cities, towns in terms of the milk marketing. So it has to be extend their availability of the products in small cities and the towns. It has to be considered the lack of efficient of cold chain distribution network. So from the production point of the industry, either to the consumer retail point, so there is a chain of distribution of cold chain network. It has to be till it reaches to the consumer table. The product has to be maintained in cold chain, and these products of our Indian it has to meet the international global standards. For that, small further level, it has to be increase the pressure on land resource for cultivation, lack of good quality animal feed, lack of animal health care facility, low genetic potential of animals. Like in morning class, Dr. Vikas sir has told about the genetic potentiality of the animals. It has to be produced, it has to enhance our local breed, their productivity enhancement, the lesser to the greater extent. And also lack of chilling capacity and the proper veterinary practices, it has to be followed, should not be have the final products of either the antibiotic residue or the veterinary drugs at the final products. So the collection level, it has to be depends on the small holders and make the involvement too many intermediaries and also the lack of infrastructure and manipulation of quality of milk by the farmers. So mainly the alterations is the point where the quality is also get deteriorated. And the processing level, milk in India, the flush season and lean season and winter season. So whereas in case of flush season, we may get abundant of milk production. Whereas in case of lean season, there is a deficient of milk production. So we need to be balance this seasonal imbalance of milk production. So for that, the processing level, it has to be start to meet these the fluctuation of the supply. Absence of quality standards with respect to the raw milk quality or the finished products, it has to meet the national and international standards. And we should need to take care about the alteration and the food safety standards. Lack of trained and skilled workers are need to be trained so that they can handle the milk and milk products at the farm level or at the processing level or at the marketing level. Storage and logistic level. So it is already been mentioned that we have to enhance our cold storage facility, and we need to fulfill the gap in cold chain and transportation facility. So cooperative level, less number of members of the member farmers, and lower participation in the decision making process, low price of milk, inefficient services, inefficient infrastructures are the main cause for the decreasing the quality of milk in our cooperative level. And there are certain marketing challenges where the majority of the market milk is still in the need to be at the organizing sectors, acceptability of the customer base, less penetration to the rural market, and lack of transparent milk pricing policies. So these are the challenges are there in our Indian dairying. But there are certain strengths of our Indian dairy is as the milk has reached to 198 million metric tons of during the present 2022. In that, the increase of the purchasing power of the consumer. So earlier, it was only focused on the fluid milk. Now the trend has been changed to the quality of the products, the nutritional benefits are also the consumer also aware about the nutritional benefits of the products, and also improved transportation facility for movement of milk and milk products. There is increase in the availability of indigenously manufactured equipment is also available in our country. Large number of dairy plants in the country, and also the availability of trained and qualified manpower is also been available. So this is not sufficient. We need to be enhance the infrastructure, the manpower, and in all the areas of the Indian dairy industries. Weakness of our dairy is lack of appropriate technologies. So we need to be transfer these technologies from the developed countries like the New Zealand, Netherlands, Denmark, and all other. They are handling the products online. Starting from the raw materials to the finished products, there is no intervention of the manpower. So and also the erratic oversupply. So this is also one of the major weakness of our industry. Then lack of awareness on the hygienic production, and also the regional imbalance of milk supply. So apart from that, so India is to focus on the export potential of milk and milk products to the Western countries, and expanding the market of traditional dairy products, increasing demand of fluid milk as well as the value-added products within the country. The country is having a huge market for the marketing of milk and milk products. So we need to generate the employment generation and the availability of large sources of unconditional feeds and fodders. So these are the strengths and the weakness of our Indian dairy industry. Certain success story or case stories I would like to share with you, that one in Tamil Nadu Milk Unions, especially the Madurai district milk union, Salem, Eero district, Philippines. There we had implemented the clean milk production. Before implementation of clean milk production, the bacterial count it was about 40 to 50 lakh, which is inferior in the milk quality. After the implementation of clean milk production, by washing the udder and the area and the cleaning of the animals, after the implementation of clean production practices, that the bacterial load it was 10 to 15 lakh. But the same society level, once the bulk cooler it has been installed, the bacterial control count was drastically reduced to 3 to 5 lakh of the bacteria. So from this, where the bacterial the bulk milk coolers are being installed, we can install the high or superior quality of milk. So another success or the case two is the Hassan Milk Union, which is comes under the Karnataka Milk Federation. There during 1996, the plant was 120,000 lakh liters per day. Now it is handling 380 tons lakh liters per day, and it was expanded to 50 lakh liters per day. So it contains 3 talukas where it is handling about 380,000 liters per day. So presently, with all this, the union is handling about 1 lakh liters per day, which the bulk milk coolers are being installed to 37 bulk milk coolers, and also the automatic milk collection unit, it is about 403. So as of this, the sales of the marketing of the products, it was raised to 2.5 lakh liters, and curd it was 50,000 liters, and other products like butter, ghee, buttermilk, peda, and UHT milk. The ice cream is having a separate processing unit of UHT milk and ice cream unit, and also the flavored milk, which has been recently there is a one separate unit which has been packed in headquarters of flavored milk. So these are the success are being due to the improvement in the milk quality at the farmers level or at the village level. So there is certain ongoing project is also there, the fully automated mega dairy, which is been for the whey protein concentrates. So as we are all aware that the success story of the Amul Anand Milk Limited, the taste of India. See, before the success, this Amul, it was we were all knows about that how this was the Amul. It was been succeeded. Earlier, it was been the Paulson dairy. It was a private dairy which was been exploited for the farmers, by the middlemen and the traders. For that, government has made a scheme, the Bombay Milk Scheme, to purchase the milk from the farmers, and it will transport to the Paulson. Paulson, it is again it will transport to the Bombay milk colonies. So, but this was not the need of the farmers of the Khaira district. So for that, they have formed a society during 1942 under the chairmanship of Tribhuvandas Patel and the Sardar Vallabhbhai Patel. So for that, during 1946, you all know that the Dairy Khaira District Cooperative Milk Producer Union Limited, which was registered during 1946, and now it is called the Amul Anand Milk Union Limited. So this Khaira district, after the registration, it has designed a 3-tier system of milk collection. So it is called as the Anand model of milk collection, which is from the village milk which has been collected, and it has been transferred to the milk unions and then marketed through the federation. So this is how the 3-tier system of Anand milk cooperative system has existed. Excuse me. So the model which has been replicated throughout the country as a model, and under the to exist a setup, a board, National Dairy Development Board was existed during 1962, For that, a Operation Flood program, which is the largest food program, was launched in during 1980. So that in the face of, excuse me, it's audible. Is it audible to you?
So, yes, sir, you are audible. All the participants are
sir.
requested, please
Thank you.
mute yourself.
Yes, sir. Thank you. So, in three-phase manner, Operation Flood Program 1, 2, and 3. So, the mainly focus is on the whatever the commodities are available in the European Union, the butter oil, the milk powder, through the free of cost of World Food Bank. So they brought to the India and they converted to the fluid milk, sell the products of fluid milk to the metropolitan cities, generated the revenues. They start constructing the infrastructures. They're constructing the dairies, providing the veterinary services, making the crossbreeding policy of high yielding animals with the local indigenous breeds. Then the providing the training the to the farmers. This is how the Operation Flood program was succeeded throughout the country. So India, before this Operation Flood program, India was severely deficient of the milk. Now it is in the stand of importing the products to the global. So in this way, we should thankful to the AML to make the India a #1 milk producing country, and the products are also exported to the many countries. So in that way, the total turnover of the AMUL is 61,000 crores, and it is reaching to 30,000 villages, and it is almost 3.13 millions are producers members are there, which is the largest brand in India with respect to the dairy industry is concerned, and the 18th largest dairy organization in the world. So in this way, it is a proud moment that the Anand Milk Limited, it has been made its own stand of brand, and it is a brand model for the countries of the other industries to reach the international market. So future prospects of Indian dairy milk production is expected to increase around 330 million tons in next another 10 years. So increases the milk supply to exceed the demand of 38 million tons in another 10 years. So plan to increase the organized milk handling to 41% next year to 50% in another 3 years. Milk procurement by cooperative is expected to increase to 20% in 2023 from 10% in 2020. And also the private sector role in procurement is projected to 30% by 2023. So these are the future prospects that the most of the milk which is been in the hand of the unorganized sector, either in the vendors or in the which is written in the village level, this it has to be increased in the hands of unorganized sectors. So from this, I'll go for the conclusion that first, the cooling facility at the village level, it has to be make the cooling within 20 to 2 to 3 hours after the milking. Under the cold chain, starting from the farmer's production, the plant which is been processing, and at the dispatch the products, and it reach the market to the retailing point and at the consumer, it has to maintain the cold chain till to the reach to the consumer table, and it has to be adopt hygienic practices like good hygienic practices, good manufacturing practices by implementing the clean production practices, and also the installation of automatic milk collection unit, the installation of bulk milk coolers, and the milk it has to be to make the chill meet the cool at the village level, which is been less than 5 degrees centigrade. And also the productivity enhancement of indigenous breeds has to be taken care. Then the final is the strengthening of the infrastructure. So it has to be, because the during the operation program, what we have is strengthen our dairy industry. Still, it is existing. It has to be strengthen the facility and make the products in the intervention of the without intervention of the human manpower. So this is how we can reach the globally international standard with our Indian dairy products. From these few words, I would like to conclude my presentation. Thank you.
Thank you very much, sir, for your very excellent presentation. You have covered all the aspects related to whatever the future present scenario of quality of milk production in India, and whatever the sources of milk production, then HACCP concept, whatever the challenges for strengthening Indian dairy industry, and then you have even mentioned the success stories, future prospects of Indian dairy industry, and whatever the requirements for the clean milk production, whatever the hygienic practices may require. So that is very much important. And as well as as you said that the production of indigenous breeds is very important, as well as strengthening of infrastructure related for the clean milk production. And we have to definitely maintain the cold chain while transporting the milk to the retail market. So thanks a lot, sir. Now 2-3 questions from the participants. If you have questions, you can ask. So if no questions, then we'll thank the participants as well as the speaker, Dr. Devraj Naik, sir, for your excellent presentation.
Thank you, sir.
Sir,
Yeah.
sir, I am
Yes.
having one
Yes,
question.
please.
Sir,
Please
I'm
introduce your name.
Dubey.
Yes.
Dr. Rajput Dubey from Indira Gandhi Krishi Vishwavidyalaya, Raipur. Sir,
Yes, sir.
am I audible? Am I audible, sir?
Yes, sir, it is audible.
You are
Yes.
audible.
Sir, my question is, as we have seen that about 60% is the unorganized sector as far as Indian dairy industry is concerned. So, is it possible to convert this at least 60-40 ratio to 50-50 or 70-30? Is it possible?
Definitely, it is possible. One is Ashutosh. One is earlier it was the transportation problem, the road facility, the infrastructure facility. Now government is providing a lot of the financial facility to the strengthening the infrastructures. So in that way, is definitely the industry, the sector, the organic sector is looking forward to collect the milk where the milk is not been collected. It is opportunities there to go for another sector also.
Sir, but it may also change the milk consumption pattern.
See,
For
that is
example, like,
the consumptions is not, it will not be disturbed, but it is the point of collection. Either the middleman, the vendors might have role in that area. So that is the reason they will not sell the bulk milk.
okay, thank you. That's what I want to know. Thank you.
One more question is there. Is there any SPC standard for Indian curd?
Standards, I need to check, sir.
Question is there in the chat box, and the participant can directly ask the question. he is mentioning that is there any standard plate count standard for Indian curd?
So I need to check. Definitely for every product there is a standards are there as per the Food Safety Standard Authority of India. So I'll check it.
Good afternoon, sir. Myself, Maske Sachin, PhD scholar from Warner College of Dairy Technology. I'm audible, sir?
Yes, it is audible.
Sir, my question is, what is the effect of cold plasma in future prospect of milk processing? Cold plasma is a non-thermal technique for milk processing. It is absolutely is from ICT Bombay. Is used the cold plasma for fruit and vegetable meat and processing. What about the dairy processing, dairy product and milk product about in cold plasma processing?
Cold plasma, I'm not aware of it.
Can you elaborate the question? What exactly you want to ask?
Sir, actually, cold plasma is a technique, non-thermal technique for preservation of food and food product. It's also used in ICT Bombay for the meat processing, and then also used in fruit and vegetable processing like strawberries. What is the now he used it for milk and milk product?
As a what that ingredient is added in that product?
Yes,
Okay.
sir.
Well, plasma, huh? Plasma
Yes.
is added as a what? In foods.
Plasma is a bombardment of plasma therapy and plasma made from fraction of electricity and doses of the
So far, the technology is not been adapted right now. So definitely the research in future days, if it look forward.
Yes.
Okay,
Yes.
sir. Okay.
Actually, it
Yes,
is
sir.
novel non-thermal food processing technology. I think
Yes, sir.
it is mainly used for energetic reactive gases to inactivate contaminating microbes on the meat, poultry, foods. But now
Yes,
it is
definitely, sir.
not yet much popularized, you know,
It's not much popularized.
Okay, so we need to
Yeah.
have
Thank you, sir. Thank you so much, sir.
yeah.
Yes. Is there any question? Okay, then if no questions, I am reading the chat box. It is your lectures are quite impressive because many participants they have mentioned that excellent information, nice, very nice information. So that indicates that you have given the updated and very recent information on your talk. Thank you very much, sir. Once again, thank you very much.
Thank you. Thank you to everybody. Thank you, sir.
And yeah, thank you. Regarding tomorrow's program, that is there is a rescheduling of lecture. Instead of Dr. ZB Dubal at 12 o'clock, there is his lecture was there. But instead of that, Dr. C. Latha will present her talk. Because of that, she is busy on 23rd as there is a Vice Chancellor's meeting uh, at her university. So that's why she has requested to prepone her lecture. So tomorrow, Dr. R.S. Kambavi's lecture will be there and Dr. C. Latha. So then thanks to all the participants and the speakers for today's program. And we will meet tomorrow at 11:45. Thank you very much.
Thank you, sir.
10:45.